Okay, so the topic given to me is infections and immunity and vaccines. So I'll try to divide it into two halves. We'll talk about infections and immunity in the first half, maybe take two questions in between, and then go on to vaccines. These are my disclosures. So yesterday, our co-chairman, sir, Sai Atikul Haq, sir, he mentioned that Mishra sir was like the Incredible Hulk. So in immunology in India especially, he is the one of the pioneers. He is the teacher of teachers. He's the teacher of Danda sir, and thankfully is my teacher also. And the giants have already spoken. Mr. Sir has spoken, Danda sir has spoken, and even great scientists like Dr. Saha, Dr. Abro did a wonderful job. So my job is very easy. Can you see me on the screen? I'm there. Compared to
Yes,
this,
yes.
that is where I am. Maybe Ant-Man. So the topic for first topic is infections and immunity. Most of the work of immunity in infectious disease has mostly been done in primary immunodeficiency, which will be covered in the talk coming after this one. Then some amount of work has been done for HIV, tuberculosis, parasites. Professor Saha has done some work, but now what we have seen in the COVID-19 pandemic is that we cannot afford to neglect immunology in infectious disease. So coming from the Marvel Cinematic Universe to Shakespeare, this is from the play Hamlet. In Hamlet, he's told his friend Horatio that there are more things in heaven and earth than are dreamt of in your philosophy. So we know only very superficial things about immunology, especially when it comes to infection. We are barely scratching the surface, and there is a lot more to learn. You people have asked wonderful questions. Sometimes it is very difficult to answer them, and hopefully we have been able to satisfy some of them. So to start, I'll talk about antibacterial immunity. Nothing new. All the major topics have already been covered by the previous speakers. I'll just introduce the same things from a different perspective. What happens when the bacteria enters the body? So we know innate immunity starts from physical barriers, physical as well as chemical barriers. The skin has different molecules, some are called defensins and many others are there, which do not allow bacteria to grow on the skin. Then no bacteria can penetrate intact skin. Even our mucous membranes, they have a layer of mucin, and which has again defensins and other innate molecules which prevent the entry as well as the growth of bacteria. Even bacteria which can penetrate intact mucous membrane are very few. Mostly the Corynebacterium, couple of Neisseria species, Haemophilus influenzae, and Listeria species. So, how do the bacteria enter the body? They can take a help of something which is known as a vector. Vector, suppose a mosquito is biting or a mite or a tick is biting, they can help introduce the bacteria or the parasite inside and cross the barrier. Or otherwise, there has to be some other like smoking or environmental pollution, which is disrupting these barriers and making us susceptible to various bacterial infections. A big thing coming up is the microbiota. Did you know that there are 10 times more microbial organisms on our body than there are actual cells? If we have 10 to the 8 number of cells in our body, the number of the various microbiota, be it bacteria, fungi, virus, which live in and on around us, on our skin, in our gut, in our nasopharynx, everywhere, they constitute 10 times more if we look at the number of cells. And these also help to shape our immunity. One repeated question was coming, that how our immunity can improve. Immunity is never a static thing. It is in a constant flux. It is changing. Every time we eat something, few bacteria will go to the gut. It will change the gut microbiota. When we brush our teeth or pass the stool, there is some microaberration, some bacteria is going in. The entire immunity of those barrier surfaces can change, leading to profound changes. So it is very difficult to say what influences immunity. Actually, everything does. but nothing can be uh, nothing can be permanent. What are the defenses? Once the bacteria has entered the body, penetrated the barriers, they have to face phagocytosis. There can be neutralization or blocking by antibodies. Some natural antibodies can be there, and some preform antibodies because our body has seen that infection previously. Complement activation can be of two types. It can be innate activation also, or it can be activation occurring via the acquired immune system. But what is most important is that there is a need to recognize the pathogens. So how the pathogens are recognized? So these are recognized with the help of patterns. Suppose we look at these two shadows. We can say possibly this is a man and a dog. We don't know who is that man. It may be a girl also. We don't know the age of the person. We don't know the name. And this may be a dog, this may be a wolf. It may be of various species, we don't know. But from the distance, when we see this shadow, most likely this is a man and a dog. So this is a pattern. Similarly, the immune system recognizes patterns. What patterns can be recognized? Like lipotecoic acid. Lipotecoic acid is normally not present in mammals. Then certain lipoproteins, or like methylated DNA. Methylated DNA will not be present in prokaryote, in eukaryotes. It is present in prokaryotes. So if these foreign molecules are there, that means something is wrong. And these molecules are called pathogen associated molecular patterns. Molecular patterns, as the name suggests, it is just a pattern, and they are coming from some pathogen. So the innate immune system can recognize that this is foreign by looking at these kind of patterns. And the recognition of patterns is by pattern recognition receptors. What we have heard, TLR, they have been discussed. So if there are certain like zymozyme that is present in fungi, or lipopolysaccharide which is present in bacteria, there are different receptors which can recognize this, and these all receptors are non-specific. It doesn't matter what is the structure of the LPS, and these are mostly lipids or sugars and not proteins which have different tertiary structures. So we can divide these pattern recognition receptors as those present in the surface and those present intracellularly. So those present in the surface are more important for bacteria which are outside, extracellular bacteria. Most of the viruses we see, they will be intracellular. And there are certain PRPs which can detect the viruses which are inside. Suppose single-stranded RNA should not be there. dsDNA should be in the nucleus, but not in the cytoplasm. Double-stranded RNA should not be there in humans. So if this type of molecules are coming, this can be picked up by the receptors on the innate immune system. Also, the complement system, the Mannose binding pathway and possibly the alternative pathway, they are also part of the innate immune system because they can non-specifically recognize. Suppose mannose. Mannose is a sugar which has a 1 to 3 alpha binding, which is not present in mammals. So if mannose is there, that lectin gets activated, and that mannose binding lectin, it initiates the complement cascade. Then there are various others. We call them TLRs, RLRs, NLRs, CLRs, and they have different functions. TLRs we have seen, RLRs are mostly intracellular. Then NLR, I'll just mention because NLRs have certain importance. Suppose this is a schematic we can see. There are various bacteria on the surface. They are leading to certain molecules which are coming inside the cell. And these are what we call the PAMs, or pathogen associated molecular patterns. They will activate the node-like receptor, and node-like receptor leads to a beautiful structure which is known as inflammasome. The various node-like receptors, they will come together, they will polymerize. You can see this structure. The three-dimensional structure is much more beautiful. You can see how beautiful the structure is. This is the three-dimensional structure, and this is called an inflammasome. Inflammasome because it initiates the inflammation. This is very important. Initially, it was discovered in Mediterranean fever. Then people saw it was there also in gout. Now people have understood that this is there even in coronary artery disease. You cannot get atherosclerosis without inflammasome activation. So what we have learned in a rare disease, a primary immunodeficiency or autoimmune disease that has come to infectious disease and that has gone into various other diseases. That is the beauty of immunology. And what this inflammasome does, it takes a molecule which is pro IL-1 or pro interleukin 1, and as the molecule goes through this three-dimensional structure, IL-1 or interleukin 1 comes out from the other side. And this is IL-1 is a very potent immunomodulator, or it starts the immune reaction. So when there is detection of the bacterial products, it leads, if it is digested, and it leads to the immune reaction, which is started by IL-1 beta, and other is IL-18. Then coming to the second group, this have been discussed, what we call a danger signal. We can see a smoke coming out here. What does the smoke imply? Smoke implies that there is a fire going on. So this is indirect proof that something wrong is going on. So these are known as, these danger signals are known as DAMs, or danger associated molecular pattern. Suppose ATP should be inside the mitochondria. DNA should be inside the mitochondria or inside the cell. But if we are getting ATP in the plasma, or ATP is coming out in the cytoplasm, that means some problem can be there. F-actin is a cytosolic protein. If it is coming out in the plasma, then there are various molecules present in the nucleus, like the HMG, high motility group B1 or N1. So if these molecules which are normally present in the nucleus are coming outside, then something has to be wrong. So this can be regarded as the smoke coming from the fire. Some cells are being destroyed, maybe by a virus, maybe by a tumor, maybe even by injury, and that is leading to release of these molecules. And these also can be recognized by the innate immune system and lead to an immune reaction. So putting it together, first there has to be breach in the mechanical or physical barrier. Then first line, there is always some natural antibodies or the complement activation which will attack. Then the phagocytes will come, the neutrophils will come, the macrophage will come, they will try to engulf and eat up the bacteria. And meanwhile, the bacteria will release some toxins so it can invade further. And toxin neutralization is again another important feature. Mostly, since it is extracellular, this is mediated by the antibodies. Then in case of certain infections, there is also role of the mast cells. We'll look at that. So the bacteria has its own attack mechanisms like attachment, proliferation, invasion, toxins, flagella. All these are the bacterial attack or their weapons. And to counteract each of them, the body has a different mechanism. This is just a schematic video, not actually, but you can imagine this is what, these are the receptors on the bacteria which can come and help attach to the human cells. But the antibodies are coming and attaching to those receptors. Suppose we take the example of the spike protein of the block. The virus cannot attach to the AC inhibitors and be internalized. Then this slide was shown yesterday also, but because of the questions, again, I'll repeat it. How does the complement system, the terminal complement system act? That trigger for the terminal part of the complement is C5b. What C5b is does is it cleaves C6, C7, C8, and a lot of C9. And all these C9, they form a polymer. Again, this polymer forms a pore. And this is the electron microscopic picture. This is the three-dimensional schematic diagram. And here we can see on the right side that these pores are being formed. And once these pores are formed, all the material inside the bacterial cell will come out and the bacteria will lyse. And this is most effective against envelope viruses and gram-negative bacteria. Gram-positive, but the bacteria are also clever. What they do, they have a capsule. So once they have a capsule, the phagocytes are not able to engulf them. The pattern recognition receptors are all hidden inside the capsule. Even if the complement will try to complement will not get activated because of this protection. We have such like Streptococcus, Klebsiella, Escherichia, Neisseria, Haemophilus. So these are encapsulated organisms. So, and what do we need to do? We need to make them attractive for the phagocytes. So like putting sauce on the samosa. So the bacteria are being opsonized. Opsonization can be done if the complement goes and binds or if specific antibody binds. So it is like putting markers so that the phagocytes can see and engulf them. What is important for this group of bacteria is the spleen. In the spleen, the microcirculation is very rich in WBCs. And the splenic capillaries, if you remember, they are around 4 to 5 microns. And RBCs are bigger than that. RBCs are usually 7 to 8 microns. So imagine any cell which has to pass through the splenic capillaries, they have to go single cell at a time, and all the white cells surrounding that splenic sinusoids, they are able to scan each of the molecules if there is any opsonized entity. And this is very important for getting rid of the encapsulated bacteria. So what is the clinical implication? If a person has asplenia or person has had splenectomy because of maybe hemolytic anemia, person had splenectomy, or in case of sickle cell anemia, there is autosplenectomy. So they are very much susceptible to what is called opportunistic post-streptococcal, it should be opportunistic post-splenectomy infections or OPSI. So this can be a very fatal. The fatality rate is 40 to 50%. And when we are going for an elective splenectomy, it is always recommended to vaccinate for all the encapsulated organisms. As I showed, the spleen is important for them. Neisseria, yesterday it was mentioned, people who have terminal complement deficiencies, they are much more susceptible to Neisseria meningitis and other Neisseria infections. Then the complement receptor tree can be used as a, because the bacteria are also clever. So they try to use parts of the immune system itself to attack the body. So one example is the complement receptor tree, which are used by these three fungi as well as bacteria to enter the host cell. So when do we get increased risk of bacterial infections? If there's a phagocytic defect, most common neutropenia. Person has severe infection or because of antibiotic associated neutropenia, or rarely because of some primary immunodeficiency, person may have a neutropenic problem. And the number of phagocytes are not there, they are not able to engulf the bacteria. Complement deficiency, there is increased susceptibility. More commonly, if there is an antibody deficiency or when there is a B cell defect, we can get bacterial infections, we can get fungal infections. Mostly the extracellular defense is lost when there is B cell defect or antibody loss. And we also talked about splenic function loss, which may lead to life-threatening OPSI infection. The DAMP has been covered. This was I audible for this encapsulated organisms?
Yeah.
Yes. Yeah.
So encapsulated organisms we have covered. Then risk of bacterial infections.
Yes, you have mentioned also.
Okay. Then the question comes whether that since antibody is important for bacterial infection, does IVIG or immunoglobulin from outside help in sepsis? So this was a meta-analysis done from the Cochrane library. And they have very high level of evidence. They do the meta-analysis and systematic reviews very thoroughly. And what they have seen in the, they have taken only studies with very low risk of bias. And these studies have shown no benefit of IVIG, even IgM enriched, or in adults or in children, they have different subgroups. There is no benefit. So possibly the amount of antibodies in the IVIG is very less. Suppose there is we are giving 5 gram of antibody, but against one bacteria, the amount of specific antibody will be very tiny, very minute amount of that. And in case of the human body, it is very different because B cells can produce tons of antibodies, and that is only specific antibodies are being produced. So this was about bacterial infections, and coming to antiviral immunity. Antiviral immunity again can be of two types. There is some role of the humoral while the virus is entering. It can be blocked by the various antibodies, and there is also, as we have seen, some role of complement. And much more important is the cell mediated, because viruses are intracellular. and two molecules, one molecule is interferon gamma, and the cytotoxic T cells and NK cells, which are again cytotoxic. So they have a role. So what is more important is cell-mediated immunity. There is contact killing, as it was discussed in the last Saturday, Sunday classes. The cytotoxic cells, they come in contact with the cell which has the virus or the tumor cells. Then they uh, extrude a ligand, which is called a fast ligand. And when the fast ligand attaches to the receptor, it induces death via death domains in the cells. Other method is when they can inject two molecules, one is perforin, other is granzyme. So this, when injected into the infected cell, they lyse and are eliminated. And one thing which is very important for viral infection is interferon. Interferon comes from the word interfere. So they interfere with the viral entry, they interfere with viral replication, and as well as the viral release of the maturity and release of the viron particles. So we can see when a cell is infected, it will release the interferon, which has a paracrine, autocrine effect also, and also it activates the immune cells. This is just to show the pathway. What is most important is that important in the interferon pathway are the JAK and the STAT, because JAK STAT inhibitors are coming in a large way. We have tofacitinib, we have baricitinib, and many others are coming in the pipeline. So we should know that these have a high incidence of viral infections. The possibility of viral infections will go up. Also, this interferon is important in mycobacteria. We'll just touch upon that. Coming to COVID also, people have shown that the interferon is important. There are two studies published in Nature Science Journal that was in October. These two studies are there. The first study has shown that persons with inborn errors of immunity, if they have some certain polymorphisms in genes associated with interferon, like interferon response element 7 or the interferon alpha receptor 1 or TLR3, then they have a chance of developing life-threatening infection as compared to people who have asymptomatic infection. So this is a genetic defect which makes persons susceptible to COVID. Another paper published in the same journal, they have shown that there can be acquired defect also. So there are antibodies which are being produced against type 1 interferon. How these antibodies are being induced by the virus, whether this is a epiphenomenon, it is not known. But persons who have autoantibodies against interferon, the amount of interferon in the blood goes down, and these people are also having increased risk of life-threatening infection. This is not in all the patients. 10% of the patients, they have autoantibodies against interferon, and maybe another 10-15% have some genetic defects. So it is not usually not one, it is usually 2 or 3 or multiple factors which leads to susceptibility of infections. Then this will be dealt on later in the next topic. If there are T cell defects, because T cell defects will affect both antibody as well as cell mediated immunity. So they lead to what is called severe combined immunodeficiency disorders, or SCID. In SCID, SCID usually have T cell. Because of the T cell defect, there is also secondary B cell and antibody defect. So there is severe immunodeficiency. In case of B cell defect, the cell mediated immunity is spared. Then again, I talked about JAK inhibitors, and certain T cell targeting drugs may also predispose to infections. Interferon defects, we have seen the example of COVID-19, where these defects can predispose to infection. And usually we expect the CMI, cell-mediated immunity, to be more effective for viral infections. But one exception is rituximab. Rituximab is a blocker of B cells. It blocks CD20 and reduces the antibody levels. But it is also important that it is important to understand that rituximab can reduce the risk of reactivation of virus, especially the hepatitis virus, and very rarely it can also cause reactivation of JC virus. This leads to a encephalopathy which can be fatal. This is very, very rare, but anyone using rituximab should be aware of this rare complication. Coming to antimicrobacterial immunity, this is more related to antiviral. This is like antiviral immunity because this is again intracellular organisms. To keep it very simple, we all know that TNF alpha is important for the granuloma formation. Then we have heard something of IL-12. IL-12 is responsible for what we call the Th1 immunity. So that Th1 cells, as we know, are important for immunity in tuberculosis. Then interferon, we have just seen it is important for intracellular organisms. And this is one gene which is again related to interferon. So most important for mycobacterial infection is TNF alpha, interleukin 12, and interferon gamma. And if we go into details, this is the same slide, but with more details. There are so many receptors. Yesterday, CD40 ligand has been mentioned. Then there is the NADPH mechanism, which is used to kill intracellular mycobacterium. This GP991 is a part of the NADPH oxidase. So if we go into details, we get so many factors which can lead to susceptibility to mycobacterial diseases. And if these diseases can be inherited in a Mendelian pattern, and then they are known as Mendelian susceptibility to mycobacterial disease. Again, this will be dealt with in detail in the next topic. When we should think of this, when a person has recurrent mycobacterial infection, he has developed TB twice, thrice, he requires therapy for a long time. That 6-month therapy is not enough. We have to go for 1 year, 2 year of therapy. Or if there are presence of atypical mycobacteria, when this type of infections are there, then we have to consider Mendelian susceptibility mycobacterial diseases, or we have to think of other causes, maybe even steroids or uncontrolled diabetes, which is leading to this atypical presentation. CMI, targeting drugs, and also we need to remember the biologicals. JAK inhibitors, if you look at the trial data shows there is no increased risk of tuberculosis. But if you look at the mechanistic view, there should be increased risk because it is blocking the JAK interferon pathways. mTORs are also coming up. They are used mostly in mTOR inhibitors are sirolimus, everolimus. Those are being used even for cardiology. Now they are using in stents, then routinely used for transplant. So these may lead to increased susceptibility to mycobacterial infection. Again, anti-secukinumab, secukinumab, that is anti-IL-17, we don't have data, but it appears maybe it is safer. And rituximab possibly doesn't lead to susceptibility because this is more B-cell related. Antifungal immunity, I'll be short. The important ones are what we call dectin, then there is TLR, these are again pattern recognition receptors which recognize fungal elements. Let me give you an example. So Candida has what we call glycan. Glycan has a 1-to-3 bond in the sugar. Most of the mammalian cells, they will have a 1-to-4 or 1-to-6 bond, if you remember our biochemistry. So if this bond is different, then this can be a PAMP, as we talk, pattern, pathogen associated molecular pattern. Similarly, Aspergillus, it also has glycan. Cryptococcus, it has mannose and certain polysaccharides. So these are not normally found in human, and presence of this can alert the immune system that something is wrong. So what leads to susceptibility to fungal infection? IL-17 blockade is important. IL-17 helps in maintaining the structural integrity, and there secukinumab that fungus is usually a big organism. It has to bypass the structural integrity inside. One interesting disease is there, episode. I don't know if Sujata will be covering it, but here what is found is that they have anti-IL-17 antibodies. So because of anti-IL-17 antibodies, they have a susceptibility to mostly candidiasis, and others obviously T cell defects, and some certain defects like card9. Card9 again is a innate cell which is downstream of dectin. So this will lead to susceptibility to various fungal infections. Anti-parasite immunity, again I'll just show one slide. For anti-parasite immunity, more of type 2 response is important. We'll just go over type 1, type 2, type 3. But what happens especially in the gut when there is a parasite, when there is a worm, there is release of the mast cell releases histamine, and that histamine causes contraction of the muscles, the smooth muscles, and that causes expulsion of the worm. So it is not just only phagocytosis and all, it can also take help of the muscular system. And because the type 2 immunity is involved, there is amount of fibrosis. Type 2 is associated with allergy and fibrosis. So in case of suppose schistosomia, we can get liver fibrosis, we can get fibrosis in the bladder, and many of the parasitic diseases we can get allergies. So the biggest question of them all, what determines the immune response? How does the T cell decide, should I go for a type 1 response, type 2 response, or type 3 response? First thing is that innate and adaptive immunity are not separate. Within the body, they are always going hand in hand. It is not that when phagocyte is there, the B lymphocyte will not be there. B lymphocyte is already there, and as soon as the phagocyte engulfs, it will present to B lymphocyte, it will present to T lymphocyte, and everything will go on spontaneously. The response may take time to mature, but it starts almost spontaneously. And this is a classical paradigm we had learned in medical school. One is the Th1, mostly the interferon and IL-2 mediated, which leads predominantly to cellular immunity. And they are important for maybe tuberculosis, other autoimmune diseases. While the Th2 leads to release of cytokines like IL-4, IL-5, and they are much more important for atopy, allergies, as well as antifungal or not antifungal, anti-parasitic immunity. But if we look at the immune picture now, we get a lot of arrows pointing everywhere, and obviously it is very easy to get confused. Let us keep it simple. To keep it simple, we can talk of Th1 we already know, Th2 we know, and we can talk about the Th3 arm. So what has been seen is that the Th17 cells release IL-17, IL-22 in response to IL-23, and these are responsible for defense against extracellular fungi bacteria, and they help to maintain the, suppose the gut immunity. They maintain the structural integrity. So this is type 3 immunity, and some organ specific immune disease, in suppose in case of Eng-spawn, it has been found that they have a role. So this has already been discussed previously by Dr. Abro. So it has a very wide spectrum. I'll not go into details, but what we are seeing, all the innate cells, the NK cells, the mucosa associated cells, the double negative cells, the innate like lymphocytes, all these gamma delta cells, these are mostly the innate cells. The innate cells are secreting a lot of IL-17 and also some of the acquired. So IL-17 is much more associated with the innate immunity. And the fourth arm, if we like, it is not actually called the fourth arm, but the very important arm is the tolerance generating or anti-inflammatory mechanisms. Because so whenever there is damage, there has to be repair. So a central cell is the T regulatory cell. The T regulatory cells helps to eliminate the autoreactive B cells, all the cytotoxic T cells. After their work is done, the regulatory T cell stops them. And it also releases certain molecules like amphiregulin, which is responsible for the growth of the endothelial cells, as well as the gut, the goblet cells and the epithelial cells. And it also leads to some amount of immune suppression such that the immune response is controlled. Otherwise, if the inflammation goes on, it will keep on causing collateral damage. Then finally, what gives the direction into T1, T2, T3? This is a dendritic cell. Dendritic cell because it has a lot of podocytes which are extending outside like dendrites of a neuron. So what the dendritic cell does, this is a plasmacytodendritic cell. Depending on the PEMP, it will have a different response. Suppose it is sensing a bacteria through TLR4 or 5 or through TLR-DIS, they will secrete predominantly IL-12 along with presentation. So this is the antigen presentation occurring. This is signal 1, and there will be one signal 2, that is co-stimulatory molecule. This is the co-stimulatory molecule, and this is the third signal. Third signal is in the form of cytokine, And this cytokine is telling the naive T cell, you turn towards Th1. So if there is bacteria is there, ultimately it is going into a Th1 phenotype, which is releasing interferon and responsible for type 1, which is effective against it. Similarly, for type 2 immunity, helminth is responsible. Helminth will have different PEMP. It will trigger different TRP, and it will lead to IL-10 or possibly IL-10, which leads the naive T cell to become a Th2 cell, and this will release IL-4, IL-5, IL-13, which leads to the type 2 type of immunity. Similarly, for the fungus, it will be a type 3 immunity where mostly IL-17 is responsible, and if there is a lack of signal or there is a signal with certain molecules like TGF beta, then the response is the naive T cell becomes tolerogenic, or it becomes immunosuppressant cell, which is known as a Treg. So this is I'm calling it the fourth arm, or the tolerogenic arm, or the immunosuppressant arm. So to summarize this, there has to be, our immune system is so perfect that uh, for every attack for the bacteria or the fungus or the microbe, there is a different mechanism to halt it. So there has to be certain defect, because no one is perfect. Everyone's genes are different, everyone's environment is different. So to have a disease, the immune system has to have a defect. It may be immune, it may be congenital, or it may be acquired. We have seen it in a case of tuberculosis. We are all exposed, but only few of us are developing tuberculosis. If we have seen in case of COVID, so we need to understand this further. And obviously, as I said at the beginning, we have just scratched the surface. Pandemic, I'm just showing here that there are much bigger pandemics which have occurred previously. Even if we compare HIV is much bigger. HIV has killed 25 to 35 million till now, while COVID has killed only 1 point, not only, but still comparatively less. So throughout the history of mankind, different pandemics have risen, and they have killed a major chunk of humanity. And to be prepared for the next, we don't know where it is coming on. So many new emerging infections are there throughout the world. So we need to have more emphasis on understanding the immunity to predict as well as prevent future pandemics also. To summarize, innate and acquired immunity work hand in hand, and all pathogens, they have different mechanisms to escape immune response, and there is always a fight going on, and host susceptibility plays a major issue not only in autoimmune diseases but also in infectious diseases. And the key cell we have all talked about since the first day, that is the dendritic cell. That is the most powerful antigen presenting cell, and that gives the direction whether this is a type 1, type 2, type 3, or an immunosuppressant. um, mm, it gives the ultimately it gives the direction, and of course our knowledge is very preliminary. So I've finished the first part, but I think I have taken a lot of time. So should I take any questions now, or should I go to the next part?
It's better to go to the next part.
Okay, so if the attendees can bear with me, so jumping into the next part. Next part is dealing with vaccines. I'll be fast. I'll just introduce the concepts. So there are so many vaccination schedules and strains, and if you do a MRI of my brain, you'll possibly find something of this size. So I'm very bad at remembering schedules and which strain is for which vaccine. So please don't ask me very difficult questions. I know you are a lot of intelligent people are there in the audience. I'll just give the overall what we call the concepts. So the basis of the vaccination is that whenever we have a second exposure to an antigen, the response is much faster, it is much larger. So in case of active vaccination, we try to tune the immune system so that it can go directly to the second order rapid and good response. I think all of us know the difference between active and passive immunity. I will not spend time on this. You have this slide on your handout. You can just see that. Passive vaccination is important in an acute phase. Suppose a dog has bit me and I do not want to get rabies. So how can I prevent? I can take antibodies against the rabies virus, so the virus cannot replicate inside my body. The number of cases where passive vaccination is used is comparatively less, and we can see them on the list here. You know this person, he got COVID, then he got a special mixture of various monoclonal antibodies which were directed against coronavirus. So he took off his mask and he said, okay, I'm protected against COVID now. But the problem is that this is passive vaccination only. He has not got a vaccine. He has got a cocktail of antibodies, and these antibodies, they will disappear from his body within maybe a month, and he will be again less susceptible maybe to future infection. So in an acute stage, sometimes we use we use passive vaccination using antibodies, but much more stronger is active vaccination. So for active vaccination, what we do, we can use inactivated virus or a bacteria, and we can use a part of it. Let us see with examples. Suppose we want to introduce the antigen into the body. How can we do it? Suppose for COVID, what the Serum Institute has done, they have taken the COVID virus and they have cultured it and produce a lot of the virus. Then they will just put a chemical in it so that the virus becomes weak and cannot infect. And that they will inject. So that is the strategy followed by the Serum Institute. This is easy to do, but it is risky because a large amount of virus has to be grown. And the laboratory standards, the BSL 3 facility all have to be maintained. Other thing is that the virus that is known as a weakened or an inactivated virus. Other strategy is attenuated virus. For an attenuated virus, we do a culture of the virus, passing the virus again and again, or even a bacteria again and again through culture, 5 times, 6 times, 10 times, weakens the virulence. The virus or the bacteria loses its virulence and it becomes attenuated. Many of the virus vaccines or the bacterial vaccines used today, that most of them are attenuated strains. Then what is the alternative to doing so many serial cultures? Because the cultures, it is a tedious process, it takes time, and there is chance of contamination. So what we can do, if we are interested only in one protein, we can grow the protein differently and then inject that protein. That is called a subunit vaccine. Nowadays, recombinant technology is there. We can produce insulin in the E. coli. Similarly, any protein we want, we can produce in E. coli or in E cells using genetic engineering. So if you want, suppose a vaccine against a toxoid or vaccine against a toxin or certain subunit, this can be done using recombination technique. Another concept is conjugated vaccine. Suppose there is a polysaccharide. Suppose Haemophilus influenzae or certain bacteria has a polysaccharide, or typhoid, the VI antigen, that is a polysaccharide. Normal polysaccharide will not lead to an immune response. The polysaccharide has to be conjugated with a protein. Then only it will lead to the immune response, and we can have an active vaccine. So this strategy, where the polysaccharide is combined or conjugated with a protein or similar large molecule, this is known as a conjugate vaccine. And these are known as haptens. Haptens are small molecules which have an immune response only when they are attached to a larger carrier like a protein. These are the licensed vaccine, most commonly used vaccines all around the world. As I said, most commonly used in the world today are the live attenuated. They have been passed to serial cultures, maybe 20, 30 cultures also, and those strains we are using. And they can be killed. We can grow in culture, we can kill them, then use them as vaccines. We can grow the bacteria, we can separate the toxoid, and that can be used as a exotoxin. Or we can use a certain protein, grow it in E. coli or yeast using genetic engineering, and that leads to a subunit vaccine. And in case of Haemophilus influenzae, as I said, that subunit has to be conjugated with a protein or some other adjuvant. These are all the licensed vaccines which are in use. Then I'll just touch upon the newer strategies. These are upcoming. People have been trying for tuberculosis, malaria, and AIDS, these strategies, but till now, none of these have been licensed for general use. One idea is we can use a nucleic acid. Suppose if we use a protein, the protein will not stay in the body for long. So possibly if we use a subunit vaccine, some may have long efficacy, but some may not have a long efficacy. But just imagine if we insert the DNA into a cell and the DNA is inside the cell, so that DNA will keep on producing the viral protein or the bacterial protein. And every time the immune system sees this, it will start a response and it will not forget this antigen. So possibly there is a chance of having lifelong immunity against this. It can be a DNA particle or it can be an RNA particle. The main problem is how to get it inside the body. Theoretically, it is better because it is there. It will keep on producing the protein, the antigen. But the main problem is how to get it inside the human cell. So they put it on a plasmid, they transinfect and put it in a carrier vector and then try to inject. So for that, we need a viral vector. Sometimes some of the vaccines, they are giving directly DNA or RNA, or sometimes they put it inside a viral vector and they try to infect. Then what kind of viral vectors can be used? That can be a replicating viral vector. Suppose we take the vaccinia virus, which is not harmful to human, and we put a new gene in it and put it inside the human being. So the vaccinia virus will replicate, but it will not cause disease. And this new gene will be expressed, leading to immune reaction and the efficacy of the vaccination. But theoretically, there is a risk because this is replicating. So some people would like to use a non-replicating, like adenoviruses, they will not replicate inside. Certain adenoviruses, they will not replicate inside the human body. The Oxford vaccine we have heard for coronavirus, that is a non-replicating viral vaccine. So they take a adenovirus 5 and inside they have put a coronavirus spike gene and they are injecting it. So it is like a Trojan horse. Actually, it is the opposite. Instead of a virus is carrying some protein or DNA of another virus. Suppose the vaccinia virus or the adenovirus 5 is carrying the DNA of coronavirus inside the body. Again, there are possible theoretical disadvantages. Suppose the person is immune to the adenovirus. We have heard that a person taking the Oxford vaccine has developed transverse myelitis. So possibly he already was immune to adenovirus and that caused the ATM. Initially the vaccine was halted, then it has renewed. Then again, what if the adenovirus goes and causes disease in the future, some unknown disease, we don't know. So another strategy what people are trying is you use a artificial virus. The artificial virus is called a VLP or a virus-like particle. So you just nowadays the engineering is biotechnology is so strong, they can make a viral code with nucleoplasmid as well as spike proteins or whatever is required, and there they will just insert the DNA or RNA and they can just put in the protein on the surface. Suppose the spike protein can be put on a nucleoplasmid and just given to the person. So these are the newer or upcoming strategies. Passive vaccination, we know we are giving preformed immunoglobulins or monoclonal antibodies. These are used only for acute situations or for immunocompromise. And active vaccines, the classical are the whole cell vaccines which are killed or attenuated, subunit vaccines including toxoid, the conjugate vaccines, and what we just discussed, the newer strategies.